So you have the next question.
Water loving.
Now,
Is that
the previous
okay?
question we said with
You
appropriate
understand, you
diagrams.
understand.
So you
Alright,
must
so
draw
let's move
diagrams.
ahead.
It's very, very essential you draw diagrams. Now, the next question you have here say, explain with appropriate illustrations the fluid mosaic model of the eukaryotic membrane. Now, in 1972, a man called, two men, actually Singer and Nicholson, postulated a theory of membrane structure called the fluid mosaic model, which is now widely accepted. Now, when you say something is a mosaic, it is actually an assortment of different things. Are you getting it now? When you have an assortment of different things, you say this is a mosaic. And have assortments, different things, you say this is a what? A mosaic. So a mosaic is a structure made up of many different parts. A mosaic is a structure made up of many different parts. You see how I'm giving you the note. This is how you should write the exam. A mosaic is a structure made up of many different plants. Likewise, the plasma membrane is composed of different kinds of macromolecules, which I have stated. The macromolecules they contain are the phospholipids, integral proteins, peripheral proteins. Now, these two here, they are combined called proteins. So we have two types of proteins, which we are still going to know very soon: the integral proteins and then the peripheral proteins. And then we have the glycoproteins, of course, and the glycolipid, which I said they form the what? The carbohydrate component. However, they are actually conjugated carbohydrates with either lipids or proteins. And then of course we have cholesterol. Cholesterol form the lipid components also. So basically there are three components of the cell membrane. We have the lipid, we have the proteins, we have the carbohydrates, and these are the three components. So if you want to actually repeat this after me, you would say that we have three components, which are the what? The lipids, the proteins, and the carbohydrates. Then they are subdivided. Of course, you have the proteins. We say we have two types of proteins, which we are going to know very soon: the integral proteins, and then we have the peripheral proteins. And then the carbohydrate components are made up of conjugated compounds, like the glycoproteins, where you have carbohydrates actually conjugated with proteins, or glycolipid, where you have carbohydrate conjugated with lipid. And then of course you have the cholesterol, which we said the cholesterol in our previous lecture actually helps in maintaining the fluidity of the cell membrane and the structural integrity of the cell membrane. That is the work of the cholesterol. And of course we said the phospholipid is like the main something in the cell membrane, right? It forms a bilayer which actually regulate what goes into the cell and what comes out of the cell. Shall we say that again? Said a mosaic is a structure made up of different or many different parts. Likewise, the plasma membrane is composed of different kinds of molecules, like the phospholipid, the proteins, carbohydrates. Under the lipids, we have the phospholipid and cholesterol, and then the glycolipids and glycolipids. Then the carbohydrate component, I told you that the carbohydrate component is usually carbohydrate, either bonded with a proteins or conjugated with what? A lipid. So you state all that. These assortments of different parts is what actually makes the mosaic. It's what makes the what? The mosaic. Do you understand that? So the mosaic is an assortment of the different lipid, the different proteins, and the different carbohydrates. You've stated that. Now, what brings about the fluid part of it, the fluidity? Like we said that these phospholipids, my dear students, we said these phospholipids you can see here is actually composed of two important parts. We have the hydrophilic head, which is the polar group, and then the hydrophobic tail, which is the non-polar group. Now, this hydrophobic tail is made up of lipids, specifically fatty acid. It's made up of what? Fatty acid, my dear student. It's made up of what? Fatty acid, my dear student. Now, this fatty acid is a complex mixture of both saturated fatty acids and unsaturated fatty acid. It's made up of two fatty acids, saturated and unsaturated. And of course, my dear student, you know that the difference between fat and oil is one solidifies at a particular temperature while one is liquid at a particular temperature, right? So the temperature difference is actually what differentiates between the fat and the oil. Now, for something to be fluid, it means it should be in the liquid form. So this fatty acid, which are saturated and unsaturated, at normal cell temperature, the cell has temperature, and of course, you know the temperature of the cell will also be approximate or can also be linked to the temperature of the body, right? Will it be room temperature? No. Temperature of the cell I can't really verify this. I'm just saying this from common knowledge that the temperature of the cell will be equivalent to the temperature of the body, because we said that the body, the cell is the basic building block of all living organisms. From the cell, you have the tissues, you have the organs, you have the system, and then you have the organism, which is the human body. So when we talk about the cell temperature, we should be looking at close to 37 degrees Celsius. All right, exactly, close to 37 degrees Celsius. Now, my dear student, at normal cell temperature, these fatty acids are liquid. They are what? Liquid form. So at the end of the day, it means that this hydrophobic tail will be what? Liquid. So you have a liquid where other things like the proteins and the carbohydrates will actually hang on top like icebergs in the sea. So the sea is the liquid form, which is the what? The hydrophobic tail. Then you have icebergs. Have you seen those sea plants where they will be floating? Ice is less dense than water. So you see ice floating on top of the water. So you have a fluid with the integral proteins, peripheral proteins, and the glycoproteins, glycolipid, cholesterol. They are floating on top, and that is what they now call the fluid mosaic model. Is that okay? Told you where the mosaic is coming from because of the assortment of different components, which include the lipids, the proteins, and the carbohydrate. The fluidity is coming from the fatty acid that makes up the phospholipids, the hydrophobic chain or the hydrophobic tail of the phospholipid, which I said at normal cell temperature is liquid form. So what we are expecting, my dear student, is this is the phospholipid. All right, this is the fluid mosaic model. If they say draw the fluid mosaic model with appropriate illustration, explain, this is what you are expected to draw. Are you getting it? You can just make a caricature of it. You must not draw a very beautiful portrait, just a caricature. You are good to go. Actually labeling appropriately and then giving appropriate illustrations, giving good explanation as to why this thing should be here and why the other should be the other way. So like I told you, of course, if you see this thing, if I'm asked to draw this thing in the exam, I can easily draw it, right? How do I start? I start with drawing my heads. So I will have various heads. These are the various hydrophobic heads. Of course, you can see them, plenty hydrophobic head. And then you now have the tail. The tail, they must be 2 in 1. So I have my tail here. This is just something you can draw very well in the exam. And then we have another tail. Can you see how it is now? It's very easy to draw. And then you have the heads, 2 tail to 1 head. Have you seen it now? So these are the hydrophobic head, which you are going to hydrophilic head, which you are going to label. So you see this region here, they said this is the what? Hydrophilic region. Under here too is the what? Hydrophilic region. And then this bilayer is the hydrophobic region, and this hydrophobic region is fluid like. Is what? Fluid like. And you see every other thing will be suspended on it, like icebergs floating on top of the sea. Is that okay? Use that word, it's very very important. Like icebergs floating on top of the sea. Is that fine? So these ones you have here, just darker color, they are what? Cholesterol. So you label this as cholesterol, which are what? The other kinds of lipids. Then I told you that we have carbohydrates like the MHC, major histocompatibility complex. They help in cell to cell communication and cell signaling, especially in immunity. All right, in cell immunity you make use of these glycolipids. Is that okay? And then what you have here is the glycoproteins. All right, now the glycoproteins, we have two types of protein generally. We have the glycoproteins, and then we have the let's say peripheral proteins, and then the forget about the glycoprotein. Glycoprotein, they will fall under which category? Under the carbohydrate part of it, right? They have their own function. But what I'm interested in are the integral proteins and the peripheral proteins. Now you can see the peripheral proteins. You see they are just on the surface, the periphery. All these ones, they are just on the surface. This one, they are called the what? The peripheral protein. Now there are some proteins that actually, you see, they sojourn the two parts of the bilayer. When I mean they sojourn, I mean they travel across the two parts of the bilayer. They are called integral because they are inside, passing through. Say, this person is insider, is integral part of the whole something. Are you getting it now? So this is the integral protein, especially this one that actually sojourn the entire length of the fluid mosaic model of the cell membrane, is called the transmembrane protein. Transmembrane protein. Now, why do you call it the transmembrane protein? The reason why you call it transmembrane protein is because it actually goes across the two sides of the bilayer. Now, what are the functions of these proteins? Now, these peripheral proteins, they generally act as receptors. All right, they generally act as what? Receptors and enzymes. Enzymes and receptors. What are receptors? These are components that actually does what? They receive certain things, like it could be ligands, it could be chemicals, and so on and so forth. You are going to learn about receptors later, like drugs. When you take drugs in your body, most drugs binds to what? To receptors. Are you getting it? Most drugs binds to what? To receptors. Is that okay? Then you have the other ones, like the transmembrane protein. They act as what? Ion channels. That means they are used for the transportation of nutrients across the cell membrane. Is that okay? Transportation of nutrients, transportation of various ions, and so on across the cell membrane. And other polar compounds like carbohydrate can be transported, because those polar compounds cannot be transported through the lipid bilayer, because the lipid bilayer is non-polar. So if you want to transport something polar like water, like glucose, you have to pass through the through the channels. Are you getting it? You have to pass through the channels. Is that okay? So that is basically what the fluid mosaic model is all about. So when they ask you to explain the fluid mosaic model, what are you going to say? You just basically explain everything that I've given to you here. Then you add your own little, Like I told you, when you write the phospholipid, you write the function. Integral proteins, you see the way I explained them, right? You write their function. Peripheral proteins, write their function. The glycoproteins, glycolipid, write their function. Cholesterol, you write their function. I've stated their functions during the course of the lecture. If you know you cannot actually put it together, you can just go to ChatGPT, type different the fluid mosaic model. Is going to give you the different components of the fluid mosaic model and then each of their function. But the most important thing is for you to understand, so that when you are given the privilege to write in the exam, you will not just write out of point based on what you learned from ChatGPT. That is the essence of this lecture. Is that okay? And then you draw the structure. This structure is very, very important because you're going to say with illustration, explain the fluid mosaic model. So you use this illustration to explain, and then of course we have the question here. Plasma membrane is made up of, it is composed entirely of lipids. Is that true? No. Mainly made up of proteins. Is that true? No. Mainly made up of lipids and proteins. Exactly. Composed of only carbohydrate and lipid. That is not true. They are mainly made up of lipids and protein. Like I told you that the carbohydrate component of the plasma membrane is minute. Alright? The carbohydrate component is minute as compared to the lipid and the protein component of the plasma membrane. As a matter of fact, you are not going to see, if you look at this structure here, what you see here is, you see the phospholipid bilayer is covering everything, but the one that have the highest mass, that's why I said by volume the lipids are much, because the bigger structure, if this is like a cake, you see that the bigger chunk of the cake is made up of lipids. Then you have all these proteins that are actually that sojourn, just some of them hang around, while some sojourn the entire length of the proteins, of the phospholipid, or of the bilayer or the lipid bilayer, right? But the components that have the highest mass is the protein. This protein, you see them here, they are small, they are not that many, but they have the highest mass because protein has higher mass than what the lipids themselves. But by volume, the lipids are much. By mass, the proteins are what much. And of course, we have minute quantity of carbohydrate, which are equally combined with proteins and what? And lipids, as in the form of glycoproteins and glycolipids. So that will be all for this lecture. Thank you very much for your time. We'll see you in the next one.